Hi and welcome to my OCR A-Level Biology revision session with me, Christine. So today's lesson I'm going to look at proteins, which is part of your biological molecules module, which is module 2. So amino acids then are the monomers which come together to make the macromolecule, which is our proteins. So you need to know the general structure of an amino acid. So here you can see my general structure. It has an amine group, which is nitrogen and 2 hydrogens, and it has a carboxyl group, which is the carbon, 2 oxygens and a hydrogen. So all amino acids have the same general structure. There is one area which is differing, and that is called the R group. So the R group is the variable group, and there are 20 different amino acids. Some have hydrophobic side chains, some have hydrophilic side chains, and some have intermediate side chains. Now, you need to understand how they interact with each other, but you don't need to know the names of the amino acids. But there is one that should stick in your head and be a go-to when it comes to naming anything to do with amino acids and disulfide bridges. So you need to know that cysteine is the amino acid which contains sulfur. So all amino acids contain carbons, nitrogens, hydrogens and oxygens. However, the cysteine also contains sulfur. So that is a distinguishing feature between amino acids and carbohydrates and fats, because the others do not contain nitrogen and do not contain sulfur. So what happens then if we take two amino acids and we join them together? Well, this is another form of a condensation reaction, because we can take the hydroxyl group from the carbon of one amino acid and the hydrogen from the amine group of another amino acid. What we can then do is bring those two amino acids together by creating a peptide bond. So what we've got here is a dipeptide, two amino acids being held together by a peptide bond. And because we have water being produced as a product, that shows us that this is a condensation reaction. Now, amino acids are different based on those R groups. So you need to understand that we are able to separate out our proteins and our amino acids using a process called thin layer chromatography. So you should know from your GCSE chemistry that chromatography has got what's known as a stationary phase. Now, the stationary phase in thin layer chromatography is a very thin layer of silica gel. Now, that silica gel is an adhesive substance, so that can therefore form hydrogen bonds with molecules. Mobile phase is where we have an organic solvent which is able to move up through the thin layer of silica gel, and as it moves up, because it's a solvent, it's able to dissolve substances in it. So it's forming hydrogen bonds. Now, these hydrogen bonds will be with, for example, the amino acids. So when you do thin layer chromatography, you would draw a line which is with a pencil, so it does not interact with any of the pigments, which are proteins, or any of the amino acids. So you would always draw using a pencil line, because that should not form hydrogen bonds with the solvent and therefore should not move through the thin layer chromatography plate. So the next thing we need to understand is that mobile phase, that movement of the solvent, we call that the solvent front. So the solvent will move up through the plate, the mobile phase, and we will stop it about 2 centimeters from the top of the plate. Now, when we do that, we draw another line using pencil, and we call the distance between the pencil line and the point at which the solvent has stopped the solvent front. So that is important when we're measuring and working out the retention value. So you have to understand that different substances have got different attractions to the stationary phase. So if there is a stronger attraction, that's going to result in a slower movement through the stationary phase, and therefore it's going to cause the substance to separate out. If there's a weaker attraction, you're going to have a faster movement through the silica gel. So therefore, what that means is we can actually take the amino acids or the proteins or the carbohydrates, and we can actually separate them out due to these interactions with the stationary phase and their solubility in that mobile phase. And what that means is that we're able to work out this retention value based on the distance moved by the molecule divided by the distance moved by the solvent. Now, most of the times, these substances that we're looking at and we're trying to measure are actually colorless. So what we need to do is we need to add in a chemical that's going to allow us to visualize this colorless substance. So ninhydrin is a spray that can be used to visualize if we're looking at amino acids. Because the amino acids react with the ninhydrin, that will then turn either purple or brown, and that means we can then visualize so that we can measure that distance that molecule has moved by the distance moved by the solvent front. So this is part of your practical assessment. You will be assessed in the exam situation, so you should do it as a practical assessment in lessons, but you also will be asked to be able to do this in your final exam. So let's take a step back then and look at how proteins are actually made. So protein synthesis is the synthesis of a protein. So an amino acid can be bonded together with another amino acid to form a dipeptide. And if we have lots of amino acids being held together, then what we get is what's known as a polypeptide. Well, what determines the sequence of those amino acids is down to the DNA. So your DNA holds the genetic code for the polypeptides to be synthesized. So through the process of transcription, the DNA is copied onto messenger RNA. So if you haven't already, please do check out my video on transcription. What you will know is that each of your triplet code is called a codon, and that is corresponding to a very specific amino acid. So through the process of translation, that code is then translated into the polypeptide. And that polypeptide has got a very specific sequence of the amino acids based on the code that was copied. So we call that the primary structure. The primary structure of the protein is the sequence of the amino acids which are held together by the peptide bonds. And it's important that you do look at the lesson on transcription and translation so you know how protein synthesis actually occurs. So once we have our amino acids, the sequence of those amino acids are held together in the right order by those peptide bonds. What we then move on to is what's known as the secondary structure. Now, the secondary structure is important because the secondary structure is going to start the shaping of the protein. Now, the key thing to know is with every single amino acid, remember there are 20 variants. Each of them has an R group. Now, up until this point here, primary structure, we are only looking at our peptide bonds. We're looking at the peptide bond being between the carboxyl group and the amine group of adjacent amino acids. When we look at the secondary structure, we're still only looking at the carboxyl group and the amine group, but this time these interactions are not with adjacent amino acids. What this is this is interactions between further away amino acids. So what happens is interactions which are weak hydrogen bonds that form between the carboxyl group of one amino acid and the amine group of another either cause a helix shape, which is called the alpha helix, or it can cause what's known as a beta-plated sheets, which happens when the long chains come parallel to each other and it forms a pleated shape to it. So the secondary structure is caused by weak hydrogen bonds between the carboxyl and the amine group. So peptide bonds is your primary structure. Hydrogen bonds is your secondary structure. If we move on to what's known as the tertiary structure then, this is where the polypeptide is going to fold and bend, and this is due to even more intermolecular bonds and interactions, but this time of the R groups. So the differing R groups are going to cause the folding and bending of that protein molecule. So it could be more hydrogen bonds. So if there are more hydrogen bonds, so we've got our alpha helix or we've got our beta plated sheets, and then we've got more folding occurring. This is between the R groups this time, and therefore that is the tertiary structure. So the hydrogen bonds are the weakest bonds that are formed in the tertiary structure. If there are oppositely charged R groups, then what we then have is ionic bonding. So the ionic bonds will again cause this folding and bending if those R groups are oppositely charged. We have our hydrophobic and hydrophilic interaction. So remember, hydrophobic, if you haven't seen my video on water and phospholipids, then please do check that out. But a hydrophobic and hydrophilic interaction is where hydrophobic is repelled from water. So therefore the folding will result in the hydrophobic amino acids being on the inside of this tertiary structure. Now the reason for that is because most of these proteins are being made in the cytoplasm, a liquid. So any hydrophobic R group chain is going to repel away from the water and fold in on itself. Hydrophilic is therefore going to like and be attracted to the water, and therefore because it's attracted to form hydrogen bonds with the water, what you're then going to get is those hydrophilic amino acids are going to form around the outside of the tertiary structure. Now, the one that I discussed before was about the cysteine. If there is cysteine in the polypeptide chain, then what you will get is two cysteines will form what's known as disulfide bonds. These are the strongest covalent bonds which are formed between those two sulfurs which are found in those cysteine amino acids. Now, the quaternary structure has exactly the same intermolecular bonds and interactions between the R groups, but this time, instead of it being one polypeptide chain, it is two or more polypeptide subunits coming together. So there is the primary structure, which is your sequence of amino acid, your secondary structure, which is either your alpha helix or your beta pleated sheets based on those hydrogen bonds, or your tertiary structure, the folding, and the quaternary structure, two or more protein subunits coming together. So if we're looking at the tertiary structure, we therefore need to talk about the fact of what's known as a globular protein. If the protein is folding and bending in that tertiary structure, it's going to have a rounded 3D shape. And because it's got a rounded 3D shape, it's going to be soluble in water. So those hydrophilic amino acids are going to be on the outside. They're going to be close to the water because they can form hydrogen bonds. Those hydrophobic amino acids are going to be folded on the inside of the protein away from the cytoplasm away from the extracellular fluids. Now, because we're talking about intermolecular bonds and we're talking about interactions, globular proteins are sensitive to both temperature and pH. So their shape can change if the temperature gets too high. And we talk about that when we mention the denaturation of proteins. Or if you move away from an optimum pH, that is therefore going to affect that globular shape. We can also talk about a protein being either a simple protein, if it's a globular protein, or we can talk about it being conjugated. So if we have a conjugated protein, what we therefore have is a protein that contains a prosthetic group. Now, a prosthetic group is a non-protein group. It therefore has a carbohydrate attached to it. So in the form of a glycoprotein, that is a protein with a carbohydrate attached, glycoprotein. It could be a heme group which is attached. So hemoglobin you have learnt about or will learn about in module 3. Hemoglobin is made up of 4 polypeptide subunits with heme groups attached. Now, those heme groups have got iron in them. You will also learn about enzymes, for example, catalase. Catalase is a conjugated protein. It's an enzyme. It has 4 heme groups attached. So it is 4 prosthetic groups, non-protein groups, iron, minerals attached to it. So a conjugated protein is one which is going to contain an extra part, this prosthetic group, which is permanently bound to the protein to help it to function. Now, the other type of protein that you need to know about is called a fibrous protein. So fibrous proteins are not rounded in shape. Instead, they're fibrous, therefore they're long and narrow shapes. So their sequence of those amino acids, that primary sequence, actually prevents them from folding up. What you tend to find in fibrous proteins is that they are mostly insoluble because they have very many repeated amino acids. And in this case, the repeated amino acids are hydrophobic. And because they're hydrophobic, they are repelled from water. Therefore, they're not going to form hydrogen bonds, which makes them insoluble. Now, they can form hydrogen bonds, not with water. However, the hydrogen bonds that they do form are with adjacent chains. So what you tend to find is the fibrous proteins come with more than one polypeptide chain together. So therefore, it's a quaternary structure. Because you have more than one polypeptide chain coming together, those polypeptide chains are forming hydrogen bonds with each other. Therefore, this is a quaternary structure. Now, they normally have a structural role, so they're normally very strong and they're normally very flexible. So there's 3 that you need to know as examples. Collagen, the structural support in our blood vessels. These are long, strong, rope-like structures with the flexibility. So they are strong to withstand the pressure of the blood that moves through your blood vessels, but they're also flexible to allow for, as the heart is pumping and pushing the blood out, there's a bit of flexibility so it can stretch. Now, another fibrous protein that you need to know about is called elastin. So elastin, again, found in your blood vessels and also found in your alveoli. The elastin has the capacity to stretch and recoil. So therefore, it has that flexibility, that fibrous protein that is long but can be flexible to allow for it to be stretched, but then will recoil back into its position so it remains and keeps its structural shape. And another one that you need to know about is that keratin. So keratin is a fibrous protein that is found in the hair, your nails, and your skin. Now, keratin has lots of cysteine, so therefore lots of strong covalent bonds with those disulfide bridges. Now it's important to note, depending on how many cysteines are present, depends on how strong and flexible you have. So if there's lots of cysteine there, it's very strong and normally inflexible, and also it's insoluble. And if you've ever been unfortunate enough to have that smell of burning, if hair has ever been burnt, what you tend to smell is the sulfur being burnt, and that's what gives off that horrible smell when there is any type of hair that's being burnt. So they are the different types of proteins that you need to know. So how do we identify proteins then, that proteins are present? Well, to identify the proteins being present, we do that through the use of a Biuret test. So the Biuret test is where you're going to use a few drops of sodium hydroxide solution, and then you add a copper sulfate solution. So when you add these two solutions together, that is called a Biuret reagent. So you can either add each of the solutions individually, or you can have it as a Biuret reagent that you just add the drops to. Now it's important that you note that it's a negative result. It's going to be blue. When you add your sample with the Biuret reagent, you will then shake it very gently. If there is any proteins present, that will give you a positive result, which will give you a purple color. So you can see in my picture the negative result and then the positive result that you would get when proteins are present. So I hope you've liked this video, and if you have, then please do click the like button below and subscribe to my channel. Also, do check out my revision platform www.aiqchat.com to help you to revise.